Welcome to Chapter 5. And here we're going to talk about maternal effect, also known as why inheritance is not so simple, or Mendelian. So just to remind you, the null hypothesis and those 4 rules of Mendelian inheritance. These are the 4 that y'all should be thinking of when you think of Mendelian inheritance. In this maternal effect, we're going to see how that directly violates this first rule of Mendelian inheritance. So in this case, what you have in your chromosomes, i.e. what alleles you carry, may not actually be what's determined in your phenotype. So the maternal effect, and with this phenomenon, the genotypes of the father and the offspring themselves do not affect the phenotype of the offspring. So essentially, the mother's genotype determines the phenotype of the offspring. We're not talking about how well a mother gives care to her offspring. We're talking about a phenomenon that occurs at this level. So in this, the developing embryo level. So at this point, the accumulation of gene products that the mother provides to her developing eggs is what determines the particular phenotype of that offspring. And specifically, in the example we're going to talk about here, we're going to be talking about cleavage patterns that actually set the stage for the embryonic development. So the first example of a maternal effect gene was discovered in the 1920s by A.E. Boycott. And he was studying the morphological features of the water snail, Limnaea peregrina. In this species, and in a lot of gastropod species, the shell and the internal organs can be arranged in one of 2 directions. So this is a nice party trick you can do with any type of gastropod. Some are right-handed and some shells are left-handed. So what I mean by this is if I take this shell and I put it in my right hand, the aperture, or the opening right here, aperture, is actually going to be on the side of my thumb. So it's right-handed. If I take this shell and put it in my same right hand, that aperture, or opening, is on the left side, or towards my pinky. So that makes it a sinestral type of coiling. That shell pattern is mirrored by the arrangement of the organs on the inside, or the soft parts of the gastropod. So the dextral orientation is the most common, or more common, typically, and is considered to be dominant in its effect. Now, the pattern itself, or that curvature pattern, depends on the cleavage pattern of the egg immediately after fertilization. So if you cleave in this orientation versus this orientation, that determines whether you're going to be right-handed or left-handed, i.e. with the aperture on the right side or the aperture on the left side. So let's look at this cross and think about how this differs from a monohybrid cross involving a dominant allele, or the typical SMI, or simple Mendelian inheritance. So here we've got a cross, and then we've also done a reciprocal cross, and that's a way that we can actually see what's happening in this system. We're crossing true breeders, one that has the homozygous dextral condition, the other that's homozygous sinestral. If we cross these 2, the only type of genotype, not the only type, the genotype that we're going to get in the offspring is going to be heterozygous. So that means all of these individuals are going to be dextral. That makes sense. However, if you look over here in this reciprocal cross, we're doing the same cross, except we have different parents in terms of the sex assigned to those recessive or dominant homozygotes. We get the same production of all heterozygotes. However, we get a totally different phenotype, and all of the offspring exhibit the sinestral pattern. So same genotype, but different phenotype. So if we go down to the F2 generation and we cross individuals from either this mating or this mating, we get a prediction would be, if you cross 2 heterozygotes, is a prediction of a 3-to-1 phenotypic ratio. We do not see that here. Instead, we get all dextral. If we go to the F3 generation, the 3-to-1 phenotypic ratio shows up, where we have 3 dextrals and 1 sinestral, which is typically what you would expect under simple Mendelian inheritance. However, it should have been up here in that F2 generation. So what the heck is going on? Well, Boycott couldn't figure it out. Sturtevant actually later explained this, why this is incongruous, or this pattern is incongruous with Mendelian inheritance. So snail coiling is due to the maternal effect gene that exists, again, as that either dextral or sinestral alleles. The phenotypes of the offspring depended solely on the genotype of the mother. So if you look at our individuals in the F2 and even in the F3 generation, notice that the F2 generation are all dextral. Any mothers from either this cross or this cross are going to carry the big D allele, so the dominant allele. That dominant allele is going to cause a dextral cleavage pattern in the developing embryos. So in the eggs, those products, the D products, the big D products, are going to influence that pattern. If we look down here, we can explain the recovery of this 3-to-1 ratio, because in here, for every one of these 3 mothers that have either the homozygous dominant or are a heterozygote, you're going to have one female that's going to be homozygous recessive. Even though she doesn't exhibit the right-handed pattern, her eggs are going to be influenced by these little sinestral alleles. So that's how you could get a 3-to-1 pattern in the F3 generation. So let's talk about what's really happening here. So in this situation, this pattern, non-Mendelian inheritance pattern of the maternal effect genes, is explained by the process of oogenesis in the female animals. So maturing oocytes are surrounded by these maternal cells that are referred to as nurse cells. They are providing the eggs with nutrients. Since these nurse cells are diploid, they're both, in this case heterozygous, both of the versions of this allele, the products of this allele, both the dextral and sinestral proteins, are going to be produced, and that includes messenger RNA, and are going to be given to this egg. Now, if the mother is homozygous dominant, or big D, or a heterozygote, big D, little d, all offspring are going to be dextral because of the influence of that big D protein. So even though you're sending little d messenger RNAs and products into this cell, this one actually, the dominant allele, exerts the most pressure, and that'll cause that right-handed body plan. Now in this situation, it should be pretty clear. The nurse cells are homozygous for the recessive allele. Then that, because that's the mother's genotype, that will be the phenotype for that egg. Now this mother could have been a dextral, have a dextral phenotype, but all of her offspring, or all of her eggs, are going to produce this sinestral phenotype, because her genotype is actually little d. Now again, the sperm, even though the sperm is important in terms of the development of this this fertilization of the egg, the genotype of that sperm is irrelevant, because you're already determining the cleavage pattern before this egg ever gets fertilized. Now, this kind of effect in invertebrates, and especially in humans, is really difficult to identify and determine what we have, and we most likely do have maternal effect genes. However, because these types of genes typically play important roles in the early steps of embryogenesis, there's ethical issues for doing this type of research. Now, one example is found in the bicoid gene in Drosophila. And actually, they've identified several dozen maternal effect genes in Drosophila, our good old fruit fly friends. And in this, the bicoid gene actually produces a protein that stimulates the development of anterior structures in the embryo. So that would be the development of the head. Hope I'm getting my anatomy right. Whereas if it's defective, you're going to end up with two posterior ends. So two butts and no heads. So these kind of genes can have really profound effects in the sense of producing a viable embryo. Because these kind of, as you can imagine, these kind of mutations that lead to defective embryonic development are usually result in the loss of the offspring or that embryo through spontaneous abortions. And to end on that positive note, next we'll talk about epigenetics.